You're changing that. But what you're measuring on the right y-axis here is the rate of a specific reaction. So that's why it's called an action spectrum. How fast is that reaction going depending on which wavelength of light you're providing? In this case, the reaction you're interested in is the rate of photosynthesis. So this blue line is the action spectrum for a green plant. And you can see that it peaks in the blue, it dips in the green and yellow, and then it peaks in the red again. And notice the peaks correspond to chlorophyll A. So where chlorophyll A absorbs maximally, that's where the rate of photosynthesis is maximal. But there are places where chlorophyll A doesn't absorb any light at all, and yet there is plenty of photosynthesis. So these other pigments, and where they absorb light, also matter in terms of photosynthesis. So chlorophyll B is important, carotenoids are important in photosynthesis. And then you can see there are regions where none of these pigments seem to be absorbing any light at all, and yet there's photosynthesis. So that's a little peculiar, and this is where I get to tell you that absorption spectra of pigments, when they are purified in solution, is different than when they're attached to proteins. So these absorption spectra are produced by extracting the pigment from the membrane into an alcohol solution or usually an acetone solution, and then putting it in a spectrophotometer, giving it different wavelengths of light, and measuring the absorbance of light by the pigment in solution. That's not the same as what happens when the pigment is attached to a protein in the membrane. The action spectrum is for the whole intact chloroplast, usually a whole intact leaf. So those pigments are not in acetone solution in a test tube. So there are regions where your acetone solution pigments don't absorb any light, but in fact, when they're actually in the plant, they are absorbing light at much lower levels than they do in other regions where there's a peak. But there is actually some light absorption that is not shown in your purified extract solutions. So you should always remember that. Purified extracts are not the same as what happens when the pigment is bound to a protein in the membrane complex. Everybody with me? Yes, ma'am. Sure. So these peaks that are measured for individual pigments, like the chlorophyll A has a peak here and it absorbs nothing here and then it absorbs there again. That is done when you take an acetone or alcohol extraction of the pigment. So it's purified pigment in a solution. And then you put in a spectrophotometer, give it 400 nanometer light, measures absorption. Give it 500 nanometer light, measures absorption. Same thing with chlorophyll B, same thing with the different carotenoids. That's not necessarily the same absorption you would get for that pigment if the pigment were in its natural environment attached to a protein sitting in a membrane of the thylakoid. So that attachment to the protein changes the absorption spectrum of the pigment. So there are regions in this graph where the absorption spectrum shows no absorption of light, but there is photosynthesis. And that's a little peculiar. If none of these pigments are absorbing light, how could you have photosynthesis? Hold on. There are several reasons why this would be happening. One is that these absorption spectra are mainly purified pigment. The other thing is that, well, it says carotenoids, but that doesn't necessarily mean that every single keratin and xanthophyll that was present in that plant extract. Somebody's hand up. Yes, sir. The rate of photosynthesis, which is part of your question, has nothing to do with the purified pigments. The rate of photosynthesis is for the leaf. The absorption spectra are usually produced with purified pigment extracts. So they don't perfectly match. So there are reasons where the purified pigments are not absorbing light, and yet when you look at the leaf is photosynthesizing. And you say, well, if the pigments are not absorbing light, how can the leaf be photosynthesizing? And part of that is that you may not necessarily have included all of the carotenoids. That's a possibility. And the other possibility, which is probably more likely, is that purified extracts are not the same as the thylakoid membrane. Because the pigment in the thylakoid membrane is bound to a protein complex, and that changes the absorption spectrum of the pigment. And how that happens, you can actually take a look at by later on when you take upper level courses. One of the most famous experiments that looked at absorption spectra was done in the, I remember correctly, 1890s. So Engelmann's experiment. Sorry? So the way this experiment was carried out, took a sprig of algae and then shone light on it through a prism. So you separate the different wavelengths of light. So some parts of the algae are getting blue, some parts of the algae are getting green, some parts of the algae are getting yellow or red regions of the spectrum. And he also had a type of aerotactic bacteria, so these are aerobically respiring bacteria that require oxygen, and then look to see where these bacteria aggregated. So where would they aggregate? Yes, sir. I'm asking you where the bacteria would aggregate. Yes, sir. Where there's more oxygen, if they're aerobically respiring and require oxygen, wherever there's more oxygen, you should get more bacteria. So then you look at where the bacteria aggregated, and you see, okay, they aggregated here, they aggregated there, they didn't in the middle here. Then you can say, all right, the ones on the left, lots of blue light. The ones on the right, lots of red light. In the middle, green and yellow, not that many bacteria aggregated there. So that told Engelmann what? Yes, ma'am. I can hear you at all. All right, so where you have more bacteria means more oxygen, which means more photosynthesis. So this was his way of making an action spectrum. The rate of photosynthesis for him was measured through aggregation of bacteria. More bacteria meant more oxygen. More oxygen meant higher rates of photosynthesis. The highest rate of photosynthesis in the blue spectrum, in the red, lowest rate of photosynthesis, green and yellow. So far, so good? All right, so this is for green algae. This is Spirogyra, by the way, with the spiral chloroplast. Also, same type of action spectrum for green plants. Green algae and green plants have the same pigments. They have the same type of action spectrum. Doesn't work for all algae. He also did the action spectrum for red algae, like Porphyra, and for green algae, like Ulva. So Ulva, here's a picture, is sea lettuce. It's a green alga. It's 2 cells thick. Can be very big in terms of its diameter, but it's only 2 cells thick. It's paper thin. It's like cellophane. You can see through it. It's a little slimy. It is edible. So the colonists that settled the Northeast, when they first settled North America, actually ate sea lettuce as a vegetable. So you get lots of minerals out of that. It's kind of slimy. I haven't eaten it, but it doesn't look appetizing to me. But then again, Porphyra doesn't look appetizing to me, and you all eat Porphyra if you eat sushi, because that's the alga that they dry up to use to make sushi. So if you've had sushi, you've had nori, which is Porphyra. Porphyra is a red alga. So if you look at the action spectrum for Ulva, that's the same as what we saw for Spirogyra. Peak in the blue, peak in the red. Then you look at Porphyra, peak in the green. Very different. Now, what Engelmann, who was the first one to notice this for these red algae, suggested was that, well, Porphyra grows in deep water, Ulva grows in shallow water. So Ulva grows along the coast in very shallow waters. The light quality in the shallow water is the same as light basically above water. So it's lots of blue light, lots of red light, lots of green light, lots of every wavelength of light. And like plants, these green algae have peaks in the blue and the red. Porphyra grows in much deeper waters, and as you go, any of you do skydiving? All right, quite a few of you. What happens when you go down all 50 meters? Sorry, scuba diving. Yeah, skydiving is you go up. Scuba diving.
He died.
So, what happens when you go down the 50 meters deep? It gets really dark really quickly. And why is it getting dark?
Less light.
So what happens to the light as it hits the water? So the blue light gets refracted back. What happens to the red light?
It transmits deeper.
Blue light actually gets absorbed by water the red light gets absorbed by water molecules. Blue light gets refracted. The green light penetrates deep. And if you have any plankton algae above you, they also absorb the blue and the red. So the algae above you absorb the blue and the red. Water molecules absorb the red. Water molecules refract the blue. The only light that passes through is green. So when you get to deeper waters, there's plenty of green light, but there isn't that much blue or red light. So if you are trying to grow in deep waters and you have a peak photosynthetic rate in the blue and the red, you're out of luck, because there isn't much red light or blue light coming down there. But if you happen to have pigments that allow you to absorb maximally in the green, which is not chlorophyll, then you luck out, because those pigments can then help you photosynthesize in much deeper waters. And so Engelmann's conclusion was that, well, algae, like Porphyra, red algae typically, have pigments that reflect the red. So if you were to shine regular light on them, they look red. But in deep water, they don't look red because there isn't any regular light coming down there anyway. Green light is coming down, and green light is what they absorb most. And they actually photosynthesize well because they have pigments that absorb in that part of the spectrum. Well, that's nice for Porphyra. Doesn't necessarily apply to all green and red algae. So Engelmann said, well, green algae, because they have lots of chlorophyll, they should follow Olva's example, and red algae should follow Porphyra's example. In the 1970s, Ramus was, I think, the first one who documented several cases of red algae in surface waters and green algae in much deeper waters than some red algae. So that suggested that, well, this may not necessarily apply to other red and green algae. It might work for Porphyra and Olva, but it may not work for the others. So what else could be affecting the depth of water in which these algae are restricted? So some algae, like Olva, are always in the shallow waters, and other algae are always in deeper waters. So let's go to the back and then we'll come back. Yes, sir?
The type of chlorophylls or carotenoids they use for photosynthesis?
So you're still relying on the light quality. So if they had different pigments, they could survive those. The green algae all have chlorophyll A, chlorophyll B, and carotenoids. So that alone doesn't seem to answer that, because all green algae have chlorophyll A, chlorophyll B, and carotenoids, just like plants do. So that was the reason that Engelmann actually generalized and said, well, green algae should be restricted to the surface, because they all have chlorophyll A, B, and carotenoids. Yes, sir, yes, sir. All right, so maybe temperature has to do with it. Yes, sir, and sir. Oh, maybe the nutrition levels, because if you're on the coast, then you might get washout from the soil on the coast, so that would bring you minerals more so than if you're in deep waters. Yes, sir. Water pressure, if you're in deep waters, well, there's a weight of water above you, so water pressure differences might affect that. Yes, sir. The intensity of the light, not just the quality in terms of the wavelength, but whether or not you have 100 KR units as opposed to 5 KR units reaching. So how shaded you are as opposed to what wavelengths you're getting. that could matter. And actually, Ramos's conclusion was about the shaded, the green algae